So we're talking about proteins and amino acids in this chapter, chapter 16. So the function of proteins. Proteins are super important, and so they have different functions. They have different classifications. Proteins are all made from amino acids. They kind of have different characteristics and functions depending on what that protein needs to do. They form different structural components. They function as enzymes. Enzymes are just like catalysts in biology. And then they also, you have proteins like hemoglobin that transport. So when we're looking at different classifications of those proteins, we have, you would need to kind of know what the class is if I tell you the function. So you would say like, oh, here's a protein that does this. What class would that be? So your structural proteins, they help you kind of build things. So they provide structural. You're going to have like keratin, collagen, those things really help form like structural components of life forms. You've got contractile proteins, and these are like actin and myosin, making your muscles move. You've got transport proteins, and so your hemoglobins, your lipoproteins, they're transporting things. We know about lipoproteins from the last chapter. That's how we transport lipids around. Hemoglobin transports oxygen through our blood. You've got storage proteins. You've got your hormone proteins. You've got enzymes, which are catalysts, and so you may need to kind of remind yourself a catalyst is something that speeds up a chemical reaction. So enzymes are special proteins that speed up biological reactions. And then you've got protective proteins, and these are the ones that work for your immune system, and they have receptors on them that recognize things that are foreign to your body to like send other pieces to alert, to destroy this thing. It's foreign. It's not something that I want in my body. So an amino acid, you have different amino acids. I'm not going to have you memorize all the amino acids. I'm going to give you a list of them for your final exam. But you do need to know kind of how the amino acids are put together. And so you've got a central carbon. And so we can see our central carbon here. The central carbon is called an alpha carbon. And so alpha carbon is the central carbon because it's bonded to every piece. It's got an ammonium group, and so that's where your amino comes from. And then it's got a carboxylate group, which comes from a carboxylic acid, so that's where the acid comes from. So for amino acid, you've got some sort of N group, and you've got a carboxylic acid group to it as well. And you can have it protonated, meaning that you have the carboxylic acid part of it as an acid, like has an H on the end, a hydroxide, or you can have it deprotonated. In different environments, the amino acids will look a little bit different. But what you need to recognize is that you do have your amino group here, and you have a carboxylic acid group on the other side. And in the center, you've got this central alpha carbon connected to what we call an R group. And your R group is anything. And it's really important to know what that R group is, because the R group kind of will tell you the way that that amino acid is going to behave inside of the larger structure of the protein. So your classification of amino acids looks like this. You look only at your R group, and that makes sense because that's the only thing that's different for every amino acid. Everybody's got an amine group, an ammonium group. Everybody's got a carboxylate group, but not everybody has the same R groups. And so if your R groups are nonpolar, then your whole amino acid is called nonpolar. Okay, so hydrophobic, again, hydrophobic means afraid of water. So that's going to be your nonpolar stuff that doesn't want to dissolve in water. You've got your hydrophilic stuff, meaning water loving. That's your polar stuff. So take a look at these and make sure you can tell what these R groups are. You have this R group, all carbon hydrogens. Those are all nonpolar bonds. This R group is nonpolar, so we classify the whole amino acid as nonpolar. Versus this R group has what kind of functional group? Hopefully you were thinking it has an amide, hopefully. And that is a polar functional group, so we call this a polar amino acid. This would be an alkane, right? So kind of identifying those functional groups as your R can really help you identify if something is polar or nonpolar based on what you already know about organic chemistry. And you can also have an ionic side chain. So polar is like, ionic is like superpolar. So you can also have another ionic compound, like a carboxylate ion or something hanging off this side here, and that would also be considered a polar or hydrophilic side chain. So if you are nonpolar, your R group is either hydrogen, alkyl, we'll talk about that, or aromatic. So alkyl, that means that it's like an alkane group, or an alkyne, or an alkene. It's like all carbon hydrogen. So we got all carbon hydrogen stuff. So we got alkanes, alkenes, alkynes, or those are all alkyl groups hanging off. And then aromatics, remember aromatics are going to be things that have to do with benzene or other things that have resonance, right? And so you've got some other things that where you can kind of have different different double bonds kind of moving around. And so for example, here, this is an aromatic group in tryptophan, okay? Because it's got a benzene ring and then it's got some of these, this double bond hanging out, which can move around. There's a lone pair here on this end that helps that double bond move around. And you don't need to know as in detail of that. But if you do see some kind of benzene ring probably, and you don't really see a lot of like carbonyl groups, then it's probably nonpolar. And so we're looking for hydrogens hanging off. Your simplest amino acid over here is glycine. And then you've got alanine and valine as kind of your really simple ones, and then you've got some other you know, groups for those as well. But this is tryptophan, and so that's the compound that makes you tired when you eat turkey. That's that amino acid. Alright, so sorry about that. I know that was a not very great transition there. We also have polar amino acids. So your amino acid group is polar if you find a hydroxyl group, which is an OH, if you find a thiol, which is an SH, or if you find an amide, which is your carbon double bonded, your carbonyl with an N hanging off of it, and the N can have anything. And you notice that you have a few different categories of polar amino acids. We have polar neutral amino acids, and they're hydrophilic, remember, because they do like water because they're polar. And so we have polar neutral amino acids here. You may be familiar with some of these. Then we have acidic amino acids when the R group is a carboxylate. So if your R group is also part of like a carboxylic acid being a carboxylate, then it is acidic, and it's also polar, right? So you would see your kind of carboxylate, and you have another kind of carboxylate group hanging off the end of that, then it's going to be acting kind of as an acid, so we call that polar acidic amino acids. And then you have basic ones, which of course are going to have the basic functional group of the ammonium ion. And so your R group is an amine, an amine, which ionizes to give you ammonium. And so you can see these positive ammonium groups here. So your basics are going to have usually positive ammonium groups, and your acidics are going to have these negative carboxylate groups. And so this is just a summary of what an amino acid looks like. And then we also are going to give us a 3-letter or 1-letter abbreviation looking at the name. And so if you look at all of these, they have these 3-letter and 1-letter abbreviations. Okay. Again, I'm not going to have you memorize these. You might have to memorize them in future classes, though. So drawing your amino acids, they all have the same bottom part. The only thing is different is their R group. So just, I may give you the R groups, and you may have to draw like the amino acid base. But I'm not going to have you memorize all the different R groups because it's too much. But you should know that this is your amino acid base, and then whatever I tell you about the R groups, you'll just put in there. And so we'll do a quick check to tell me whether or not you think that these amino acids are polar or nonpolar. So looking at this glycine, where's your R group? Okay, well we've got, the R group is supposed to be hanging off the alpha carbon here. What's hanging off the alpha carbon? Expand it. It's really H and H, right? So what's their R group? The R group is H. groups, and that is nonpolar. Okay, what about threonine? Threonine. You've got your alpha carbon is here, and this is your R group. Is this a polar R group? Look at that. Look at that hydroxyl. That makes this thing a polar R group. Is this polar neutral, acidic, or polar basic? This is polar neutral because it doesn't have a carboxylate or it doesn't have an ammonium. They want you to draw the structure of each, but you don't have to worry about this for the exam. I will be giving you a list of the R groups. So if you can just draw, if you want to practice, if you can just draw the base of the amino acid, and then I'll give you R group information for each of the names, and I'll give you the abbreviations as well. And so here, I remember valine because it has this V shape up at the top. Cysteine has a thiol here, so if you want to try to tell me if it's polar or nonpolar, you should have a nonpolar here, and then this one is polar neutral. And then it's talking about this physiological pH, because if you're at a different pH, you become a more acidic or more basic environment, then you're going to have your ammonium can turn into an amine because you can deprotonate here if you're in an acidic environment, or if you're in a very basic environment, you can actually add an H here. So it just kind of depends on your environment where you are, if you have an actual full carboxylic acid part or in a full amine part, versus ammonium and carboxylate ion. But in a physiological pH, we see both the ions here. Alright, so proteins have different structures. We're going to talk about primary, secondary, tertiary, and quaternary structures. A peptide bond, and just putting together the amino acids, is called your primary structure. And so we're going to link together our amino acids at the carboxylate group of 1 and the amino group of another one are going to connect. And so this is called a peptide bond. So your peptide bond is that area here. And then it's named with the first amino acid as an ol, and then the last one, we're going to talk about this, with its full name. And then you would actually write the 3 letters or the 2 letters right next to each other, so you can tell me what's going on in a long protein chain in a condensed form. And so a peptide is the chain. And so if you've got 2 amino acids, 3, 4, we can call them, di, tri, tetrapeptides, right? So you can keep going up if you want to. A peptide bond is an amide bond. So look, if you see here, you have your carboxylic acid part, and then we have our amine group. And then we get that, I'm doing this backwards, we get that, you can see here, they already have it colorful, so I probably don't need to do that. They have it linked here. So the carboxylic acid, the carbonyl carbon of 1, is linking to the carbonyl carbon of this one, I guess, is linking to the amine ammonium part of this one. And you're getting this amide bond. That's your peptide bond. And so if you're looking as to, okay, what amino acids are where, look for all of your amides and cut them, like just exactly how we would do hydrolysis of an amide. That's exactly where you would cut it to kind of tell, okay, this is one amino acid and this is a second amino acid. This is a dipeptide. you may have to count how many amino acids are in a chain, and you need to know where to look, and that's where you should look. And this is a dehydration, right? Of course, we make a little bit of water over here on the side, so we'll be aware of that. And so a dipeptide, again, is going to be formed with this peptide bond. It's an amide group here, and you make some water. So if you want to practice, go ahead and pause and try to draw this dipeptide out. And so just how we would make an amide, you'd have kind of like this carboxylic acid area and this amine area, and you're going to combine them. And so you should be getting here is where you combined them. And here's where you would cut them if you wanted to break down your peptide chain. And so I can tell there are 2 amino acids here because I have 1 amide group. So when we're talking about a peptide chain, we have 2 a beginning and an end of the peptide chain. Okay, so we have the N terminus, and we look, where do we get this from? We get the N for the N terminus from, oh, here on this end is the nitrogen group, the ammonium group. And on this end, you have the C terminus. And the C terminus, you can probably guess, the end of it is the carbon, the carbonyl, the carboxylate group. And so whatever one is, when you're looking at the chain, ends with that carboxylate group, you actually don't change the name of it. But for the others, you add an O. Like it's kind of like a side chain, right? And we're just going to actually put it together. If you want to write it out with a little bit of space, it's fine because it can get a little like really big words. But alanine becomes alanol, glycine becomes glycol, and then because this is serine on the end, it's the C terminus amino acid, we don't change the name of it. But you can see here we'd abbreviate with our letters, our 3-letter code, and we have our single letter code. We can also abbreviate there. Again, you don't have to memorize those, or I'll give those to you. But again, know where are you going to cut it off. If you need to know how many letters to have or how many things to name, you're going to cut it right at that amide area there. So here I'm giving you the names of the amino acids, and so see if you can try to tell me the 3-letter abbreviation and the name. If you need to look up the abbreviations, you can. There's a chart that I posted on Canvas, and there's a chart in your textbook as well. So if you don't have a textbook, I do have a chart in Canvas. Alright, so hopefully you paused and you gave it a shot. Let's see. Here you go. Oh no, this kind of didn't work. So here is your 3-letter code, ALA, LEU, CYS, MET for these amino acids. And then here's your nice long name. We just throw everything together. If you have spaces between all of your O's, that's totally fine. We don't typically spend a lot of time saying these out loud. As far as I'm concerned, I'm not a biochemist, and so but I can imagine biochemists probably use the letters, or they will just say like ALCM for this, depending on what letter. Each of these has their own individual, like capital letter, kind of like an element. I'm sure biochemists don't run around saying this whole name. That would take forever to have conversations about long peptide chains because these things are like really, really long. See if you can tell where your N terminus and your C terminus is. We have our N terminus over here, and you have your C terminus over here. And we can also practice cutting. Cut here, cut here, cut here, so you have your 4. There's only 20 amino acids. Again, I'm not going to have you memorize them all. But we only make 11 in the body. And so the other 9 we have to eat. You have to get them from your diet because you need them to build proteins. And so we call those essential. And so here are the essential ones, the ones we don't make. We don't make histidine, we don't make lysine, we don't make tryptophan in its pure form. And so we need to get these from outside of our bodies, and so we need to make sure that we're eating all of these. If you get good protein, you will get all of your essential amino acids, but if you're trying to eat like proteins, careful if you're vegan, because it's hard to get all your essential amino acids, and so you may wind up needing to take an amino acid supplement, because if you're only eating incomplete proteins, then you're not getting all of your essential amino acids. So it can be detrimental if you're not getting all of those, because you do need those. And so make sure that if you are on a restrictive diet and you're not getting all of the nutrients that you need, especially to build the proteins that your body needs, that there are little amino they have essential amino acids and like this like soy sauce spray you can get. Like there's a lot of things you can add to your food. Do that. Just make sure you're being aware of like what kind of diet that you're eating, and then making sure that you're getting all the essential things that you need to be healthy. So we're still talking about this primary structure of proteins, and it's literally just putting together all these amino acids. It's a long chain. It's a full protein is 50 or more amino acids, and it has to have some kind of biological activity, meaning it like has to do something. But the primary structure of a protein is just the peptide bonds that hold together those 50 or more amino acids. So these are very long chains, all of these peptide bonds putting together all of these different amino acids. And so here is a tripeptide that our thyroid makes. Here's insulin. It was the first protein to have its primary structure determined. It's got 2 polypeptide chains linked by some disulfide bonds, so we see our little sulfurs hanging out here. Okay, and so we're talking about we'll talk more about denaturation of proteins, but these disulfide bonds are things we can break to denature. You have different chains, and you have your N-terminus, your C-terminus labeled here. So, I mean, you could go through and name that if you wanted to, but why would you do that? It's very long. And so looking here, if you want to use the amino acid chart to identify the answers to these, go for it and try that. This is a tripeptide. Remember, you're cutting it off here and here. And you've got your C-terminus and your N-terminus. We have endorphins in our bodies. These are polypeptides that they actually bind to receptors in your brain to give relief from any pain that you feel. They are apparently the smallest molecules with opiate activity in our bodies. Pentapeptides, so how many peptides are in those? Right, those are 5. This our pituitary gland produces hormones, nonpeptides, right? 9 peptides, oxytocin, vasopressin. They're very similar. They actually only differ on number 3 and number 8. If you would like to try to write all the combinations you could make for a tripeptide with 2 glycines and 1 alanine, go for it. Here are everything that you can make with that. Alright, so moving on to the different structures of the proteins. So, first, primary structure. Long chain of amino acids. You gotta get that chain. Now, that chain, what we're gonna do is we're gonna start condensing that chain. So an alpha helix is taking that chain and wrapping it, kind of like you would wrap something like around a pencil, right? That alpha helix is wrapping that polypeptide chain around, and it they make it akin to a spiral staircase. Okay. And how does it? It doesn't just know how to do this, right? It's doing this because of the hydrogen bonds between the carbonyl groups, and the hydrogen attached to that nitrogen group on the other side. So here's more of your alpha helix. This is your secondary structure. So remember, primary is just the chain. Secondary, now what are we going to do with the chain? We're going to coil the chain. And you can see all of my favorite little things over here. Look at those beautiful hydrogen bonds. We see we have a hydrogen bonded to an N, and then we have an O with a lone pair. It's great. And that's how we get our nice little protein helix here. And so we get this spiral staircase based on these hydrogen bonds. You also can have what we call it, you can have either the alpha helix or a beta pleated sheet. And so the beta pleated sheet, we have a different set of hydrogen bonds. And so here we get these hydrogen bonds between like the next turn, and here we get the hydrogen bonds between like the backbones. And so it just depends on where your hydrogen bonds are, so that depends on just how everything is going to be structured. But you can have either an alpha helix or a beta pleated sheet, and a pleat is like these bins right here. So it's kind of looking at this, it's kind of like an accordion bended down, it's pleated. So it gets bent down because of these hydrogen bonds are kind of pulling them together. You can also have a such thing as a triple helix, and so triple helixes are really strong proteins, are like collagen, cartilage, skin, tendons, where you have all of these like really strongly held things. We would maybe have a triple helix there. So that is another option for a secondary structure. It's all held together with hydrogen bonds. And so if you're thinking about how this secondary structure being messed up can affect you, Alzheimer's you've got these amyloid proteins existing in the alpha helical form in your brain. Okay, if you have Alzheimer's, those alpha helical forms are actually beta pleated sheets. And if you take these staircases and you make them into those sheets, you get these like, I'll show you, you get these, you're taking this staircase and you're turning them into these sheets, and then they become these insoluble things that the like water can't wash away, and it blocks the neurons' signals with each other. And so even just thinking about like going from your long chain of peptides into your alpha helix versus beta pleated, like if you're supposed to go into alpha helix but you don't, you go into the beta pleated, you get things like Alzheimer's, right, which is a big deal, right, based on something so small that's happening here, the difference between these two. So be very careful when you're thinking about this, because, you know, the difference between these structures is important to understand, because it's going to help you understand, you know, other diseases and things later on in your careers. And so we have our primary structure, our long chain. We got our alpha helix or a beta pleated sheet, or we got, you know, that triple helix. Like, we're going to put those together, those long chains, some kind of spiral or accordion sheet or braid, right? And now we're going to have another structure. So that was our secondary structure. Now we're going to have a tertiary structure. This is really where the protein is going to be formed. Okay, we're going to take all of these alpha helices and all of these beta pleated sheets, and we're going to start bending it and twisting it together, okay? It's determined by what we call the cross-links, and it's attractions and repulsions between the side chains. And so the R groups tell us the tertiary structure. The secondary structure is formed by the hydrogen bonding of the amino acid group, not the R group. All right, so based on these R groups, your structure is going to start folding. So this is called your tertiary structure. And so we have a few different types of R group interactions that can give us these different structures, but this is just an example of some that are kind of thrown in there. We're going to go over some of these. So you have a few different tertiary structure interactions, and I want you to know these. Alright, we've got hydrophobic interactions. So hydrophobic meaning water hating, and these are going to be between nonpolar amino acids. And so the R groups of those nonpolar amino acids, because remember that's how we know it's nonpolar amino acid, because the R group's nonpolar, they're going to like each other, right? Like dissolves like. Nonpolar likes each other. And so you're going to get these things that don't like water, which the whole amino acid is in water, right, in our bodies, and so they're going to be hiding from the water together, and so they're going to come together. Then you have these hydrophilic interactions between anything on the outside and the R groups of the polar ones, and so you're going to have these kind of go together. The hydrophobic chains are going to want to kind of fold inside, and the hydrophilic parts are going to want to be outside because that's where the water is. And so you kind of can think about it as like a more complicated version of the phospholipid layers that we talked about in your cell membranes. And then you've got salt bridges. So you need to get it in your mind that a salt is an ionic compound, because it's really going to help you here. A salt bridge is literally an ionic bond between the negative R group of one and the positive R group of another. That is what a salt bridge is. It is literally a bridge of an ionic compound. So it's an ionic bond. So you get a pretty strong interaction there. You do have some hydrogen bonding here. So we got a hydrogen bonding between the if you have polar R groups, the H of one of those polar R groups and the oxygen or nitrogen of another amino acid, you do have some hydrogen bonding on the tertiary structure as well. It's just not as prevalent as the interactions between the amino acids in the secondary structure. And then you have disulfide bonds, and so sulfurs will bond together, and you've got these different thiol groups specifically of your cysteine amino acids where you find those thiols. And so those two thiols can come together, and we've talked about those if you go back into the chapter about thiols. I think it was chapter 12, and the little piece of about disulfide bonds. Alright, so do a quick pause and see if you can match these. Alright, so the first one, polypeptide chains held side to side by hydrogen bonds. And so if you're looking at this, if they're held side by side, right, we're thinking they're kind of held together like this, making an accordion. That's your beta pleated sheet. Okay. A sequence of amino acids in a polypeptide chain, that's your primary structure, just your long chain of amino acids. Your corkscrew. So corkscrew is also like your spiral staircase, right? And so you've got this looping kind of thing, like wrapping around a pencil or something. That's your alpha helix. And then if you have it woven, like I'm not going to be able to draw this right, woven like a rope, then you've got your triple helix. Alright, so see if you can separate these tertiary interactions. So pause and go for it. Oh, sorry. These are, you have to see them. Sorry. If you want to look, you can at a chart. But unfortunately, they don't have these pictures for me right now. Just know that these are the answers if you want to look them up, and we can do that in class as well. Look at those and see what they are. On your exam, I will actually give you, like, oh, here's a structure, or, like, I will give you the chart of the R groups, and I'll say, like, okay, what kind of tertiary interaction is this going to have? And so I'll have the pictures for you. And then last we have the quaternary structure. And so your quaternary structure is actually when you start to get multiple proteins together. If you have 2 or more protein units, then you have a quaternary structure. And this is really what's going to start making things, doing things around in the body. It's more than one protein coming together to make this. So the hemoglobin is made up of 4 different polypeptide subunits. So to sum up, you've got your primary structure, just your amino acid chains. We're going to wind those into some kind of secondary structure, either alpha helix, beta pleated sheet, or triple helix. Then you're going to fold those into a tertiary structure and with different hydrophobic, hydrophilic interactions. And then you're going to take this and combine it with other proteins and make a quaternary structure, a big old conglomerate of things. If you're looking for things that go wrong when you're building, like, your quaternary or just the shape of your protein, one amino acid makes a normal cell into a sickled cell. One amino acid. And it causes a change between polar glutamic acid is replaced by valine, a nonpolar, and so then we're losing this hydrophilic interaction, maybe even a hydrogen bonding, and we're getting this nonpolar interaction, and it causes the whole thing to change its shape. And so we get the sickle shape just based on that one change of a polar to nonpolar amino acid. And so because we have these more hydrophobic interactions, your sickle cell hemoglobin molecules actually will kind of stick together in a certain way because they're all kind of hydrophobic. And then they're going to form these strands that do not great things in your body and can be very painful. So here's a summary of all of the protein structures. So go ahead and pause, do a quick check to make sure you know which structure that is. Here's your solution. Alright. Denaturation. Denaturation means we're going to mess with the proteins. So if you're denaturing the protein, you're disrupting the bonds in either secondary, tertiary, or quaternary protein structures. Denaturation is not breaking peptide bonds. You are not breaking the primary structure of a protein. You're only breaking, like, the alpha helix, the beta pleated sheet, or the hydrophobic interactions of the tertiary structure, or, like, which proteins are coming together in the quaternary structure. So you have a few different ways you can denature proteins. You can denature them with heat and organic compounds. They break apart the hydrogen bonds, and they mess around with those hydrophobic interactions. If you can denature with acids and bases. They breaking hydrogen bonds between polar R groups, and they disrupt ionic bonds. Because what do you think about what an acid and bases are doing? Acids and bases are going around accepting H plus and donating H plus, and so you're, like, putting H pluses where they don't need to be, or you're, like, ripping H pluses from where they need to be, and you're not going to be able to get those hydrogen bonds in the right places. Heavy metal ions, and so what I mean heavy metals, I mean like silver, mercury, lead. Those are heavy metal ions. They react with sulfurs to form solids, and so that's a precipitation reaction. You get precipitation reactions with some heavy metals in your proteins, and you get, like, silver sulfide solid falling out of the solution of where your proteins need to be together. And so if you break those sulfide-sulfide bonds, that's kind of how a lot of our structure is held together. And so that would mess with your tertiary structure. And then you have agitation. And so literally you could just, like, stir proteins too much. And that stretches their peptide chains around. Like, if you've ever beaten egg whites, then you've done that, right? You've whipped those egg whites into what you wanted to do, and you're breaking down, you're denaturing the protein with them. Also, when eggs are just straight up cooked, right? So heat is a way to do that. You're denaturing the proteins in the egg when you heat it up. If you've ever had or mixed a cocktail with egg, you are using alcohol and agitation to denature the proteins. So here's an example. Or a chart, sorry. So quick learning check, pause and read. What is similar about these two events? So they're both denaturing of the protein. Enzymes. Enzymes are really interesting. They are these proteins that allow things called substrates to come in, and then they do a reaction, and then once the reaction's over, it releases the product, which is pretty cool. They're just catalysts, and so we have studied a little bit. We talked about the speed of reactions, and so catalyst speeds up the reaction by lowering the activation energy. And so what it does is it just helps the reaction move along without the reactants having to like slam into each other really hard. It kind of makes it easy for it to react. And so you don't have to have as much energy in the reaction to get it to go. So imagine that like you're trying to get up this hill here on a bicycle, and I'm going to go up the green hill, you're going to go up the purple hill. We're going to get to the same place, but you're going to get there faster because you took a better route than I did, right? And it's going to take me longer to get up this hill. So this is a faster reaction because a catalyst makes it easier, makes it less energy to get through the rest of the reaction. And so enzyme naming, you take the reaction or the reacting compound that you're interested in, and you say ACE. Okay, and you're going to see a lot of enzyme names as you move through your rest of your studies, so you should probably learn this. Sucrase catalyzes the reaction of sucrose. So the enzyme name will kind of tell you something about what it does, or what it does, what it works with, what the compound is it works with, or what it's doing. So oxidase helps you with an oxidation reaction, or you can have some common names. So there's pepsin, trypsin, those are common names for some enzymes. And so it depends on where you're working. You may have to know some of those common names as well. And so we have different classes of enzymes, and if you look at the name of the class, you can kind of break down to see what the enzyme's going to do. Oxidoreductases, they help with oxidation reduction reactions. Transferases help to transfer things. Hydrolases help with hydrolysis. What is hydrolysis? Hydrolysis is using water to separate things. We do that when we break peptide bonds, when we break esters, when we break amides up, right? Lyases, those are adding or removing atoms to forming a double bond. Okay, lyases. Isomerases, well, they help us make different isomers when isomers are different rearrangements of atoms, right? And so isomerases just rearrange things. And then you have ligases, and I want you to think about like ligating something, meaning you're tying something together. And so ligases, they actually use energy to combine molecules. And so looking at these different categories, see if you can match these. Now here are your answers. So how does an enzyme work? Okay. An enzyme is like a Valvoline, or like a Jiffy Lube, or like a drive-through. Okay, we can go there. You drive in, the reaction happens, and only when you're done does it release you. It like grabs the substrate comes in, does its thing, turns into products, and then it releases it. So it's not like it's like kind of randomly moving things around. Like they have actual, this enzyme is specific for this substrate. And so this substrate is going to be attracted to this enzyme, and as soon as it gets close enough, we've got these hydrogen bondings, these salt bridges, these things that, you know, put proteins together. They're going to grab onto the substrate, and then the reaction's going to happen, and then the products are going to be, they're not attracted in the same way, and so the products are going to be heading out. So it's called an active site is where this is all happening. And it contains all these amino acids right at the right place that attract and bind to the substrate. But not the products, because the products get released, so it's really cool. So what you have here is you have this intermediate, which is the enzyme substrate complex, the ES. And so the enzyme is just like it's just working all the time. So one substrate comes in, reaction, products go out. Another substrate comes in, reaction, products go out. So an enzyme is just kind of chilling, and it's just doing its job all day long. So I like to think about it as like, you know, taking my car for an oil change at the drive-in ones, right? Like the Valvoline or like Jiffy Lube or something. I go in, my car, they do the work, I drive out. That's how an enzyme works. So here's an example of the hydrolysis of lactose. And so breaking apart lactose into glucose and galactose. And it kind of walks you through step by step. I'm not going to have you memorize something like this. So there's a few, there's a model for enzymes called lock and key model. That it's like, okay, the key is the substrate, and only that key fits into that lock. So like not every car can go into the Valvoline, right? Only like the one car that fits that lock can go in. And so that's a model, but it didn't really show you how the enzyme actually can change its shape to really fit a substrate, right? So it can take more substrates that are a little off from what it does. And so we have this Induced-Fit model that we use now. The enzyme is a flexible thing, and it will actually adjust to the substrate. And so you get more, with this model, you get more range of substrates that you can have that enzyme take care of. And then your shape will change to like help make the reaction go faster. And so if you have an Induced-Fit model, it's kind of what this looks like, is you have these two glucoses come in, and here's the enzyme, and it's got this kind of, you know, this shape that doesn't really fit this, but if you imagine it's kind of like a beanbag chair, and you just kind of stuff those in there, and it'll do its job and separate your glucose and your galactose. So isoenzymes are enzymes that do different reactions in the body. Iso meaning same. They're just different forms of the enzyme that does the same thing. So, for example, there are 5 enzymes that convert lactate and pyruvate. 5 different enzymes that do this. So they're called isoenzymes. If you do the same thing as another enzyme, you guys are iso, same. And so each enzyme has a certain amount of polypeptide units, and you can kind of tell where those enzymes are formed in the body based on where what those amino acids are. And so in the liver, lactate is converted to pyruvate with 4 M's, so we call that M4. In the heart, though, it's done by 4 H's. So they have different names based on what they are, and based on where they are in your body, and based on where they're formed, they're made with different amino acids. And so you can actually get information about what's happened with a patient based on the increase in certain isoenzymes. And so if you've had a myocardial infarction, then you may have higher levels of creatine kinase and lactate dehydrogenase in your body. Then you also see this, if you've actually have damage to heart, you'll see this H4 iso enzyme in your blood. And if you have the M4, right, then you would have indication that there's something wrong with the liver. So here's a little chart kind of telling you here are some of those different, let's just breaking down lactase, the different isoenzymes that do that and what part of the body that they're found. And so if you are seeing high levels of these enzymes in the blood, it likely means there's something wrong with that part of the body. Do a quick multiple choice learning check. Let's see if you got those right. And we're going to very quickly talk about these factors. So, look, enzymes work really well at body temperature, which is usually about 37 degrees Celsius in humans. If you get really slow, they're not going to go as fast, not going to work as fast, and if you get really hot, you actually start to denature it. So there's a maximum enzyme activity, there's a good temperature to get. So if you slow it down, it's going to go slower. If you decrease the temperature, it'll go slower. And if you increase the temperature too much, you'll denature it. So there's a good temperature you need to have. There's also a good pH you want to have. You can denature with pH, with too acidic or too basic. So you have your body pH around 7.4. That's your maximum activity. Too low, too high, and your enzyme doesn't work. Because all your amino acids will change their actual structure. And so like they're not doing the right, their tertiary structure is disrupted because they have different hydrophilic, hydrophobic, hydrogen bonding interactions based on those R groups can change. Like, so things get weird if you get too acidic in your body or too basic. So there's specific enzymes that work really well in like your stomach versus like they don't work really well in the other parts of your body. So here's a list of some enzymes that work in certain pH values. Of course, your stomach's going to have a lower pH for an acid, and then your liver has a really basic pH. So you want to pause and do a quick learning check here. Here are your answers. So inhibitors are the last part we're going to talk about. Inhibitors are the opposite of enzymes. Okay, so inhibitors, they mean that your reaction is not going to get catalyzed. It prevents the substrate from fitting into the active site, and it does it in a few different ways. And so you've got competitive inhibition. This is when your inhibitor is very similar to the substrate, so you kind of confuse the enzyme. It competes with the substrate for the active site. If you add more substrate in though, you can kind of overpower the inhibitor and you can make the inhibitor like not as active. And so it's not very favored when the concentration of the substrate is high. So this is showing you the two different reactions and when they're favored. And so if you really want to inhibit something, you need to make sure you have a high concentration of inhibitor to do competitive inhibition. Because if you have a high concentration of substrate, then the substrate is going to win the competition to get to that enzyme. And so here's kind of competitive inhibition. And it's a similar structure as far as the active site is concerned. And so it's keeping the substrate from coming in because it's like sitting there. Yeah. The non-competitive inhibition has a totally different structure than that of the substrate. But it binds to the enzyme somewhere else and actually changes the shape of the enzyme so the substrate can come in. So it's like a non-competitive inhibition is not like directly competing, right? So it's not like I'm going to come in and sit right in this active site. It's going to come in and sit somewhere else, but it actually changes the shape of the active site. And adding more substrate doesn't help that. So it's not like you can fix it by just adding more substrate. And so here's what non-competitive inhibition looks like. If I add an inhibitor, right, I'm trying to get this active site going, my inhibitor comes in and that actually changes the protein, my enzyme protein structure, and my substrate can't fit anymore. And adding more substrate is not going to help this because I have this inhibitor that's messing up this, right? And so it might be a good thing to inhibit a certain reaction, right? And so you need to think about ways in which that would be beneficial or maybe bad. And then an irreversible inhibition is, it just destroys the enzyme. Like, just, the enzyme can't work anymore. It usually forms some kind of bond that, like, just makes the enzyme not functional. You've got antibiotics that do this, and so penicillin does this to bacteria. They totally break down. So here's a little list of all your inhibitors. You do need to know like what the characteristics are and if it's competitive or non-competitive or irreversible. So give it a quick check. So here are your answers. And here is your concept map for 16.